All right, class. Today we're going to kick off with chapter 11 on meiosis and sexual reproduction. This is a fairly short chapter, but it is a little bit detailed. So make sure that you're making those note cards and studying all the bolded terms and concentrate on your homework quiz because that's the level of detail that I expect you to know. So, jumping right in, we're going to get into sexual reproduction. So, sexual reproduction is combining cells, sex cells like gametes, eggs, and sperm from two parents through fertilization. By mixing of the chromosomes from two individuals, the genetic diversity is increased. So that's pretty key. This is what increases diversity is mixing chromosomes from two separate individuals. And this process is going to differ from mitosis, in which one cell produces two genetically identical daughter cells, in that we are going to start out with two haploid gametes, an egg and a sperm, and they will join together in fertilization to form a diploid zygote. So that's what this cycle is showing, is that you get a sperm from the male testes, an egg from the female ovaries, and those will join together during fertilization to form a diploid zygote. So this is where the 2n, or chromosomal number, equals 46. Um, and this process of meiosis is going to vary from where plants and animals are going to have two sets of chromosomes, one set from each parent, meaning that they are therefore diploid. And then you can have a reduction of chromosomes, which is necessary to avoid doubling in every single generation. So if you didn't reduce back down to haploid sex cells, you would have a doubling of in every generation of chromosomes. So reproductive structures that produce gametes or spores in plants have cells within a single set of chromosomes, which means that they are haploid, or 1n. Okay, so here is the difference again between meiosis and mitosis. So meiosis reduces the number of chromosomes by half to produce the haploid cell, whereas in mitosis you make two identical sister or I'm sorry, daughter cells. So the process is going to resemble mitosis, but goes through an additional round of division. So you have a meiosis one and a meiosis two, where in mitosis it's just one round. So here in interphase, you're going to have a pair of homologous chromosomes in the diploid parent cell. And then you get a pair of duplicated homologous chromosomes basically meaning two sister chromatids joined to form um, a chromosome, and then you'll have this homologous chromosome or copy of it, and then that splits um, in meiosis one, so that you have a homologous chromosome separates into each haploid cell with duplicated chromosomes. So in meiosis two, you get the sister chromatids that are separated, forming four total haploid cells with unduplicated chromosomes. So meiosis is preceded by a normal interphase. You get the G1, S, and the G2 phase, and chromosomes replicate in the S phase, making identical sister chromatids. And so these sister chromatids remain attached at the centromere, and it's exactly like in mitosis, where they're connected by the mitotic spindle microtubules to that kinetochore region. And then you go into prophase one, and these homologous chromosomes pair at the synaptomeal complex, which holds them close together in synapses. So segments of these chromosomes can then be exchanged through crossing over, and this helps increase diversity. Is in this crossing over event where you have different DNA regions transferring from one homologous chromosome to the other. And then you're going to have these visible structures at the crossover points called chiasmata, and the four chromatids are held together by these chiasmata are called the tetrad. So when it's joined in this configuration here, and you see these little white squiggly lines, that those are the chiasmata, and that is holding them together. And we call this when the homologous chromosomes are joined like this, it's called a tetrad. So crossing over exchanges genetic material. We just said that crossing over event allows for genetic diversity in the species. So if you see here, you have these two homologous chromosomes, they will align, and then they can have this event of crossing over where you have a recombinant DNA being exchanged, and then you still maintain non-recombinant areas of the chromosome where no crossing over took place. But then we move into prometaphase 1, and this is when the spindle fibers and the microtubules are going to attach to the kinetochore proteins at the centromeres, and then the nuclear membrane is completely broken down during prometaphase 1. During metaphase 1, those homologous chromosomes are arranged at the cell's equator on that metaphase plate with the kinetochores facing opposite poles, and the maternal and paternal chromatids orient randomly and are mixed, and then they're going to migrate to the poles. So this creates additional genetic variation among daughter cells. So we have the crossing over event, and then we have the random orientation of migration to the poles. So that's an additional genetic variation system. So then we go into anaphase 1, and the microtubules pull those tetrads apart, and the homologous chromosomes are separated, but the sister chromatids remain attached at the centromere. So this is where it differs from mitosis. So they still remain attached at the centromeres. Then we go into telophase 1 and cytokinesis. And so we are going to separate the chromosomes, and they arrive at the opposite poles. And if cytokinesis or separation of the cytoplasmic contents into the daughter cells does occur, the nuclei does not reform. So the nuclei do not reform. And then, so here's the summary of meiosis 1. So we've gone through the cycle one time, and now we're going to go into prophase 2. So we're going to go straight into prophase 2 for the second cycle. And these events depend on species specific differences. And if the chromosomes are going to be decondensed in telophase 1, they will begin to recondense at this time. So then prometaphase 2, the nuclear envelope is going to completely disappear again, and the spindle is fully formed, and then each sister chromatid forms a kinetochore and attaches to those microtubules from opposite poles. And now this is when we start to see the process of them actually being separated during anaphase 2. So the process of chromosome alignment differs between that meiosis 1 and meiosis 2. So we see that you had the homologous chromosomes here that were pulled apart to opposite poles. In anaphase 1, they get pulled apart and separated. But in prophase 2, you just have the single chromosome with the sister chromatids connected. This is very reminiscent of mitosis at this point, because during anaphase 2 now, they actually get separated. Each sister chromatid is separated into its individual chromosome and pulled to opposite poles. So during metaphase 2 and anaphase 2, the events proceed just like it did in mitosis, and the sister chromatids separate and move towards the opposite poles. Then telophase 2 and cytokinesis, the chromosomes arrive at opposite poles, and then they decondense, and the nuclear envelopes form around the chromosomes, and you form that nucleus again. The cytokinesis is then going to separate the two cells into 4 unique haploid cells. And so here is the diploid meiosis 2 summary. So we started out prophase 2. They arranged spindle fibers connected during prometaphase 2. Then in metaphase 2, they're aligned at the equatorial metaphase plate. And then we start to see from basically here down, this is going to be very reminiscent of mitosis at this point, but with the sex cells. So this is metaphase 2, they're lined up on the plates. Anaphase 2, the spindle fibers begin to pull them apart and separate the sister chromatids to opposite poles. Once they're at opposite poles, you start to have the cleavage furrow, and you'll also see the nuclear envelopes start to surround the 4 nuclei, and you will end up with 4 unique haploid daughter cells. All right, so let's compare meiosis and mitosis a little bit. We've been doing that the whole time, but let's break it down a little bit. So both processes will divide the nucleus in a eukaryotic cell. Mitosis is a single division event. Meiosis has 2 separate division events. So homologous chromosome pairs to form tetrads in meiosis do not occur in mitosis. So we don't get this homologous chromosomal pair alignment to form a tetrad during mitosis. only in meiosis. So meiosis has a tetrad event. Mitosis does not. In meiosis, the crossing over between sister chromatids and prophase one, and this is a big thing, it happens during prophase one, and then the random assortment of chromosomes in metaphase one creates the genetic variation. So that could be a big test question. Where does genetic variation take place during meiosis? Well, it happens during crossing over in prophase one and random assortment during metaphase one. So those are the two areas where genetic variation happens during meiosis. Mitosis produces genetically identical clones. So mitosis is a cell creating two cells that are identical to each other. That's mitosis. Meiosis, four individual genetically varied haploid cells. So meiosis reduces the ploidy number by one. Mitosis does not. You get the same exact stuff in mitosis, where meiosis is reducing the ploidy number from a diploid down to a haploid. All right, and this is just a big chart that compares everything that we've just said. So you can go over this in detail yourself. It's basically exactly everything that I just said in a chart form. All right, here's study guide for chapter 11. If you're having issues with this information, there's some great resources through Khan Academy and Crash Course on meiosis and mitosis. I highly recommend the Crash Course. I'll probably email y'all those links this evening sometime. Take a look at them over the weekend. It just kind of does a really good video and graphical representation of what we just went over. But if you need additional help, please reach out to me and we can schedule a Zoom over the weekend. Your test is Monday. It needs to be done before 11:59 p.m. Don't wait till the last minute. Don't email me at 10:30 asking why it needs a code. There is no code. You need to make sure that you're, got your Proctorio extension installed correctly and done the test to make sure that it's working properly and that you don't have any kind of tabs open. Y'all know the drill by now. So no access code through there. It will be all done through Proctorio. All right. Good luck with your studying and let me know if you need any help.